Okay, so I'm going to make a screencast for chromosome mutation material just so that we don't cover it too quickly today in class. This is not material that's going to be covered on exam 3, but it is on your homework 9, so I wanted to make sure that it's there and available for you so you can finish your homework assignment before it's due today. And this is also information that we'll come back to briefly before we move on to new material ahead of the final. Chromosome mutations will be potential material that can be covered on the final, so you should definitely spend time on this. You should make sure you understand the ideas and concepts, and you're welcome to bring these to me in office hours or review sessions that are future upcoming or via email. All of that's fine. All right, so we're covering chromosome mutations at a very kind of basic level. We're thinking about how chromosomes are structured and how they may contribute to genetic variation or potential diseases that we see in humans. So we're thinking about general chromosome structure. Each of our chromosomes, when it's condensed and observable during cell cycle divisions like mitosis and meiosis, you can see the location of the chromosome centromere as well as the length of the overall chromosome. We can divide the chromosome into the small arm and the large arm. These are named small is P for petite. The large arm is named Q, which is very creatively named. It's the next letter in the alphabet. So when you're looking at a centromere chromosome that has a centromere that isn't exactly in the middle, this would be our P arm, this shorter arm here, this would be our Q arm, the longer arm here. When we have a metacentric chromosome, the P and the Q arm are going to be labeled based on the starting point and the ending point of the chromosome. You'll see P's and Q's when we talk about specific loci occasionally. So we usually refer to genes by name or by letter, but occasionally if you proceed with genetic research or genetic studies, you'll see loci referred to as the chromosome number, the arm that they're found on, and then a number that corresponds to the map units or the chromosome map position that we'll see that gene or locus. The positions of the centromere will vary and can distinguish different chromosomes. This is one of the things that we talked about, identifies a homologous pair of chromosomes, so the centromere location is the same. So our metacentric centromeres will be essentially in the middle of the chromosome. Submetacentric will be slightly to the side of the middle. As you get closer and closer to the end of the chromosome, you'll find chromosome centromeres that are referred to as acrocentric. And then telocentric are those centromeres where the centromere is specifically at the very end of the chromosome. So typically when you're trying to tell the difference between submetacentric and acrocentric, we're doing this in a comparative kind of way, looking at the entire karyotype or the set of chromosomes for an organism to make this distinction. We can also think about how chromosome mutations influence the level of ploidy of an individual. So we've talked about haploidy and diploidy. Haploidy is the case where we have half of the number of autosomes in the individual or the cell. Diploidy is where we have a 2n number, so 2 copies of all of the unique chromosomes. Haploidy in humans, for example, is seen in the gametes, so our sperm, our egg cells that are produced by male and female individuals, whereas diploidy is typically seen in somatic tissue. In general, we'll talk about a couple of exceptions to that in a little bit. But if we think about how these other types of ploidy levels correspond to what we already know, haploidy and diploidy, if we look at a monoploid individual, this is an organism that has only one complete set of unique chromosomes. We could consider bacteria cells as monoploid. They have one circular chromosome that has all of the genetic information on it. You could have one complete set of unique chromosomes where an organism has multiple chromosomes, but they only have one copy of it. These are going to typically be more commonly observed in our single-cell organisms that do not undergo sexual reproduction or the combination of gametes to get to an individual. If diploidy is having 2 complete sets of each unique chromosome, polyploid is a state of having more than 2 complete sets of unique chromosomes. So instead of having one copy from the paternal parent and 1 copy of all the chromosomes from the maternal parent, maybe you have 2 copies from one or the other parent. Or maybe your parents have multiple copies of the complete set of unique chromosomes. Polyploidy is something that's commonly observed in plants. You have some organisms that are polyploid. These are often the case, the result of hybridization events between 2 species that are technically different biological species, but they can still interbreed. And if that a hybridization event occurs, the typical process of the gamete formation and fertilization coming together will be slightly different than if you have 2 gametes of the same species. So polyploids can be indicative of recent hybrids that are formed. They're also tied to many types of plants, sometimes tied to crop plants where polyploids have bigger fruit that's produced. Polyploids are commonly observed in strawberries, for example. Those really large strawberries that you get at the grocery store, those are typically polyploid individuals where having more complete sets of each of the unique chromosomes leads to larger fruit overall. So if we think about chromosome mutations, we're talking specifically about changes that lead to variation from the wild type chromosome structure or number. So this could be the complete sequence that corresponds to each chromosome. So whether you have a complete Y chromosome or whether you're missing part of it. We talked about an example in a previous class where the SRY region was missing, which led to female individuals that had a Y chromosome. You can also have mutations that lead to the difference in the number of chromosomes that appear in the progeny, the offspring individual. These chromosome mutations are fairly common. We have some type of mutation that's detected in about 6 in 1,000 live births. We also know that chromosomal mutations can occur within an individual due to mitotic cell division mistakes. So you can have differences in the number or the kind of completeness of chromosomes in different cells or subsets of cells within an individual. And these may have an effect on the cell phenotype. They may cause the cell to die. They may cause the cell to take on aberrant behaviors. So behaviors it shouldn't have. Maybe they lead to cancers where that cell tends to grow or divide at an unchecked rate. So these can have a kind of a combination of harmless or harmful effects. Again, this level of the effect depends on the location, the size of the mutation, and how many cells have that mutation. Chromosomal mutations can be fairly severe, and a lot of times the organism in which a chromosome mutation occurs won't make it to the point where they're born. So you'll have miscarriages caused by chromosomal mutations. You may have an individual that makes it to term or a little bit preterm, and those individuals are not viable at the point of birth. Chromosomal mutations are also linked to developmental disorders. One of the most commonly cited ones is trisomy 21, where we have individuals with too many of chromosome 21. They have Down syndrome. We can also have chromosomal mutations that lead to infertility and, as I mentioned, cancer.